workflow improvement. The floor is yours, Kate.
All right. Thank you very much for the introduction, Tom. So today I'll be talking to you about our experience miniaturizing the reaction sizes for single-cell RNA-Seq using the MANTIS from FORMULATRIX. There we go. Okay, so first a little introduction to the Stowers Institute. We are a basic biomedical research facility located in Kansas City, Missouri. The Institute opened in 2000, and we now have over 550 staff members. We have a little over 20 PI labs doing all sorts of research on interesting critters like planaria, cavefish, and lizards. We also have 13 core facilities, and I am a research technician in the molecular biology core, where we do all the next generation sequencing for the Institute. So as I said, I'll be talking about our single-cell RNA-Seq workflow, so I wanted to give a little background information for that first. When doing bulk RNA-Seq on a large population of cells, the differences among individual cells are masked, allowing them to be averaged out, and the information obtained from a population average does not represent the single cell. So currently, single-cell transcriptomics is becoming very popular to study the cells individually. And to give you an overview of the single-cell RNA-Seq workflow that we use in-house, the cells of interest are first ID'd and partitioned off individually by the researcher. They are then given to our group in the core in a lysis buffer, and we generate and amplify the cDNA using the smarter technology from Takara. From that cDNA, adapters are then ligated and libraries made using Illumina's NextEra XT transposon-based method. And finally, the libraries are sequenced generally as a pool of multiple barcoded single-cell libraries. So the challenges our facility has faced for single-cell RNA-Seq include the fact that the demand for single-cell RNA-Seq projects is greatly increasing as interest in this method continues to rise. And as the demand rises, the cost per reaction or per cell becomes very inhibiting. Single-cell transcriptome analysis requires many data points, so researchers have to submit large projects consisting of hundreds of samples at least. Another challenge we have faced is that technology addressing the sample size and cost challenges has been changing rapidly, and it takes time to evaluate a method or system, and it can be hard to determine which method to go with to best suit the needs of our facility. So among our testing of various single-cell RNA-Seq methods, we decided to purchase a MANTIS robot from FORMULATRIX to alleviate a portion of the cost per sample by miniaturizing the reaction size to a quarter. So today I'll show you data as we compared two types of planaria cells. We processed 40 stem cells and 40 differentiated cells sorted on the same day by using full-sized manual reactions compared with quarter-sized reactions on the MANTIS. We used the SMART-Seq V4 kit from Takara for the cDNA synthesis, followed by the NextEra XT kit from Illumina for the library prep, as I described in the workflow earlier. We did QC on the cDNA of every single sample using the bioanalyzer, and here you can see the 40 stem cells processed manually on the left, and the 40 processed using the quarter reactions on the MANTIS on the right. Planaria cDNA is interesting. If you're used to seeing cDNA profiles, this probably looks very strange to you, but this is normal for planaria. We expect just one very narrow band as we're seeing here. So the cDNA here looks very good. We then made libraries manually in both cases. We still did full-size reactions for the set on the left, but did quarter reactions for the set coming from the MANTIS cDNA on the right. We process our libraries manually at this point just to keep our MANTIS PCR free. Here is the cDNA QC on every differentiated cell sample using the bioanalyzer, with the 40 processed manually on the left and the 40 processed using the quarter reactions on the MANTIS on the right. You'll notice the cDNA for these cells is rather varied and the QC looks pretty spotty. But this is expected. It's the nature of the cell type as they're coming from different tissue lineages. So we expect this pattern from these cells, and what we can take away from this is that the quarter reactions on the MANTIS perform similarly to the manual full reactions for the cDNA prep. And here we have the libraries made manually in both cases. Again, we still did full-size reactions for the set on the left, but did quarter reactions for the set coming from the MANTIS cDNA on the right. The libraries for these cells also show more variation than the previous cell type, but again, this is expected. Then for sequencing, we pooled all 80 samples for each respective method and sequenced as 101 base pair single read rapid runs on the HiSeq 2500 from Illumina. So now I'll go into some of the data comparing the two methods. So these plots here show the number of genes detected in both methods, and we can observe that they are very similar. Both show an average of around 5,000 genes per cell. There are a couple outliers, but those we could also pick out from the bioanalyzer QC, so we knew that going into the sequencing. These are box plots of the expressed genes for the cells in each method, and again, we see them being very uniform and comparable, very clean, and nice looking there. And when working with these two planaria cell types, we can look at a specific marker gene to determine the success of the two methods used. This gene, called PWE, should show high expression in the stem cells and no or low expression in the differentiated cells. Both methods look good here and are very similar. The differentiated cells will always have contaminants of the stem cells due to the nature of the cell isolation, so we do see a small percentage of higher PWE expression in some cases, which is expected. But in general, this is very good to see how comparable they are. Next, a list of about 60 known positive genes were sent to the bioinformatics team, and they generated this heat map to show how this data is able to separate the two sample types. On the very top of the map, you can see the stem cells shown in like a turquoise color and the differentiated cells in pink. And both methods show distinct sample separation based on these genes, with all of the stem cells clustering to the left and all of the differentiated cells clustering to the right. And lastly, we have principal component analysis showing distinct cell type separation for both methods. The stem cells especially showing nice tight clustering in both. So this was really great to see in our data quality checkpoints. So in conclusion, after seeing the data for our quality checkpoints, it's probably no surprise that our conclusion is that everything was very comparable and as expected between both methods. Getting such good results like this demonstrates to us how precise the MANTIS is, and by adding this type of automation to our workflow, we can definitely have great consistency in our results. This gives us confidence to move forward with our miniaturized reactions on the MANTIS to help process a greater number of samples at a lower cost to the researcher. Also, as more single-cell instruments come out, such as like the Wafergen Isolate and the 10x Genomics Chromium controller, which we actually have purchased the 10x instrument for very large-scale projects. A lot of these new instruments are 3' methods, so having this workflow option on the MANTIS still allows us to produce full-length transcript data for projects that may require it. Another advantage for this workflow is that besides single-cell, we also use the quarter reactions on the MANTIS for samples with low cell numbers, like under 1,000, and also very low total RNA-Seq projects. And we've had success in all cases doing this. So with that, I'd like to thank everyone at the Stowers Institute for their involvement and support in this comparison. An provided the cells, Juan, Cynthia for the analysis, my boss Anoja, and the director of our core, Karen, for their guidance.